The intent here is to give you a little bit of background in the area of gene therapy, why it is that it's a pretty hot field right now, and why it is that we feel we have a particular advantage within the Nanomedicines Network. So I'm going to cover off a bit on that, and then Christian's going to get into a bit more detail with regard to the particular projects that will be moving forward. From the point of view of gene therapy, the current status, so a bit of an historical perspective, particularly about the vectors that we've been using for gene delivery. Some recent progress, and those of you that heard my talk yesterday will understand there's been a lot of progress in the lipid nanoparticle vector area. Some applications and then the opportunities that we see for us. Now, gene therapy, what is it? I looked it up in Wikipedia, what Wikipedia said it was, and they have a good definition, I think, which is very general. Gene therapy is the therapeutic delivery of a nucleic acid into a patient's cells as a drug to treat disease. And so it's really just getting, it can be siRNA, it can be mRNA, it can be a plasmid, it can be gene editing construct, whatever it is. This is a general definition, and I think this is the definition that perhaps we should adopt within the network. Now, most gene therapy efforts have used viruses to deliver the nucleic acid. And so this is the idea, the little red square here. I pulled this off the internet somewhere. A protective gene put in a viral vector, which then infects the cell and produces the protective protein in the absence of any deleterious effect from the viral vector itself, which is actually a tall order. Now, the current status is obviously is really since about the middle of 1970s, early 80s. There's been a huge effort. We've known that gene therapy is potentially a big deal. It's quite obvious. So there's been over 2,000, I think 2,300 to 2016. Gene therapy clinical trials, and almost all of those are viral vectors, use viral vectors. That has resulted in a grand total of 4 gene therapy drugs approved by the FDA. 3 of those use viral vectors. Only 2 of those are in vivo. That's for Luxturna and for, I can't pronounce that one, Zolgensma. For muscular atrophy, spinal muscular atrophy. They're hugely expensive, $2 million a dose. And then there's one ex vivo, which is for CAR T cell therapy, which got approved quite recently. Point being that for all this effort that's gone in to the viral vector field, the field has not, there really hasn't been much, well, you can say there hasn't been much progress. There has, but it's what the areas are very niche, and they're also, as I say, very expensive. One non-viral, which is Onpattro, which was I talked about yesterday, which was approved last year, but I think is a harbinger of many more to come that use non-viral vectors. So that's a bit of background. I mean, the fact there's 2,000 gene therapy trials tells you of the interest and the money and everything else that's gone into this. These are not minor league efforts. These are huge efforts. So people are very determined to make this work, and it's a measure of the competition that we're going to see in this area. So the attraction is pretty obvious. Essentially all diseases can be potentially treated using the gene therapy approach. I know that's a bit of a mouthful, but it's true. And so most of the efforts to date in the clinical trials have been in oncology, cardiovascular, CNS, musculoskeletal, etc. But you can see that this really does go right across the board, and is going to make some of our choices actually quite interesting in terms of there'll be so many opportunities that choosing the right topic or the right direction is going to be a bit difficult. But it does open up everything. It's just it's an incredible situation that we're in at the moment. So why has the progress been so slow with viral vectors? First of all, they have a limited capacity. They can't carry that much genetic information. They're notoriously difficult to engineer and to manufacture. You can only give one dose. The second dose, the immune system recognizes it, and so second doses are not effective. You always have this potential for insertion into the host genome. There's a potential for viral transmission. In other words, maybe they produce more of these viruses and it becomes infectious. And then there's this extremely expensive aspect where, up to $1 to $2 million per dose, which is, of course, for a rare disease, usually inhibitory. Now, the knock on non-viral vectors, or vectors that do not use viruses, has been poor transfection potency. They're large and stable, and they're often very toxic, or can have been toxic. And so what's changed? And and what is our gene therapy advantage? And that is that we've developed some much improved non-viral vectors, high potency, low toxicity. And in order to understand why this is, you need to understand a little bit about cationic lipids and ionizable cationic lipids. But our advantage is we developed this technology here in Canada. I should say, though, that the world is catching up very quickly. So in 1987, cationic lipids were invented. So these are positively charged lipids, and it was found that if you made vesicles of these cationic lipids and then threw in DNA, RNA, or whatever to form these complexes, in vitro you would get at least a proportion of this, or some of this, penetrating into the target cell, and then you could see gene expression. And so as an in vitro, this is lipofectamine and others, as an in vitro vehicle, this has been very useful. But there's big problems. There's no cationic lipids in nature. If you have cationic lipids in a biological systems, they're highly toxic, which obviously restricts application. The complexes are large, positively charged, unstable, can't access target cells in vivo. So while there's been many attempts with complexes, they're universally been unsuccessful. And we invented these ionizable lipids, which I talked about yesterday, which allow you to package at low pH where they're positively charged, but subsequently bring that up to neutrality, and they're net neutral and much less toxic. And so this has been used to encapsulate the nucleic acid-based drugs, and they form small systems which are stable and have all the requisite properties to actually be a pharmaceutical. So this is they really do not only allow us from an IV point of view to circulate around and get to various tissues. In other words, avoid the immune system, which is usually pulling these things out of the circulation. We also found, and this was great good luck, that these ionizable lipids also allow us or facilitate intracellular delivery. And this has been these particles are taken up into a cell by a biological process known as endocytosis, which many of you will be familiar with. And the trick here is to get this material out of the endosome before it progresses onto the lysosome, where it gets chewed up. And so these cationic lipids allow that situation to occur. Now, yesterday in my talk, I alluded to the need to understand a little bit about membrane biology and the properties of lipids in membranes. So I thought I'd throw this slide in to emphasize what I mean. A lot of membrane biophysics is extremely useful for the design of effective systems. So on the left-hand side of this slide, I'm indicating a nanoparticle being taken up as things progress. Is that there's an inwardly directed proton pump in the vesicle, the intercellular vesicle, that reduces in the endosome, which reduces the pH below to about 5 or so. So these ionizable lipids will then be transformed from a neutral form to a cationic form, a positively charged form. And then they can interact with the negatively charged lipids there to form this so-called cone shape on the right-hand side. And there's all sorts of biophysical work that's been done here. And this really transforms the structural preferences of these lipids from a bilayer to what's termed the hexagonal H2 phase. This is basically a blob of fat and will not sustain a membrane barrier. This feature was completely unexpected, but it's turned out to be enormously valuable to engineer these lipids so that they take full advantage. So by optimizing the ionizable lipids to get maximum endosomal escape, we've so far got more than 4 orders of magnitude improvement in the potency of these systems, and we will get more as we through the network as we work with some of our chemists to develop improved systems. So this is just indicating the progression. I showed this slide yesterday, but we're certainly going to be doing better than that. So we'll extend the range of tissues. Right now we're good at the liver. We'll be going intraocular into the brain, into other tissues, and also we'll be extending like endothelial cells, etc., bone marrow. That can be transfected. The improved potency will allow us to reduce dosages. We'll be optimizing for different payloads. I showed this slide yesterday, but it's a reminder that actually what we're doing here is pretty serious. There's many people suffering from diseases particularly hereditary diseases, there's no treatment for whatsoever, and this is the amyloidosis, the transthyretin amyloidosis that Onpattro treats. And so if there's there's a high motivation for me, let's put it that way, that we're actually going to be able to make a difference, or intend to make a difference for these individuals who are afflicted with these disorders. So this is just the potential that we have for enabling gene therapy in general, silencing pathological genes with siRNA, expressing therapeutic genes with mRNA, and this is I've listed some of the diseases here. There's many others. Gene editing, correcting a mutation so that the correct copy of the protein is made. The range is huge. In the Boston area, I've been down there 2 or 3 times in the last 6 months or so, and there's literally hundreds of millions of dollars going into the companies that are starting to push in this gene therapy direction, and the same goes for pharma. So it's a very interesting time to be part of a network like this. I think we have a real opportunity to make a huge difference